Bismillahirrahmanirrahim. Good morning, respected chair, distinguished judge, and dear selection committee. I'm Abrar Hussain, a PhD research scholar from Pakistan. It's a great honor and privilege for me for being selected as a Best Researcher Award in the field of probiotics and microbial biotechnology by Global Sensor Award. My sincere gratitude to the Global Sensor Award for being selecting me for this prestigious award and gave me the opportunity to share my research with broader audience. Today, I'll be talking about exploring the next generation probiotic potential of indigenous Enterococcus strain through an integrated phenotypic, genotypic, and probiogenomic approach. These are the aims and objectives of this research work. We will select some selected enterococcal strain representing different environmental niches of Karachi, Pakistan. Then we perform their phenotypic assays. Then we perform evaluate different traits followed by their whole genome sequencing, then probiogenomics analysis, and their comparative genomic analysis. So let's start. What are probiotics? So probiotics are also called good bacteria, friendly bacteria, or beneficial bacteria. And according to World Health Organization and Food Agriculture Organization, these are the live microorganisms. When taken in sufficient amount, they give health benefits to the host. According to the International Scientific Association for Probiotics and Prebiotics, probiotics are live microorganisms that, when administered in aggregate amounts, confer health benefits on the host. Elimination cough is known as the father of probiotics. Once the proposed elimination cough proposed the concept of longevity, that if we change the gut microbiota with good bacteria, they can enhance the lifespan of microorganisms. This concept attracts researchers across the globe, and the field of probiotics has been flourished well. In 1965, Lilly and Stilwell coined the term probiotics, followed by the definition of the World Health Organization, and then the 2013, the ICCBS defined probiotics as a consensus statement, and currently this is used as the world recognized definition of probiotic microorganisms. According to their selection criteria, not all microorganisms are used as probiotics. There are some selection rules that should be followed for a strain to be claimed or to be used as probiotics, like their safety nature, their survival in the gastrointestinal tract, adhesion potential, antimicrobial properties, like immunomodulation potential, their technological properties, and their health benefits, genomic stability, and some application-based study can be selected or can be keep in mind once selected any strain or their probiotic potential. So based on this selection criteria, several strains and species from different microorganisms dominated by the genus Lactobacillus and Bifidobacterium has been explored well by their probiotic potential, and they also include some species from the genus Enterococci, although the genus is considered as a opportunistic pathogen. So our interest is to explore the probiotic potential of Enterococcus species. So Enterococcus is considered a ubiquitous microorganism. They are found everywhere in the environment. It have double phase gram-positive microorganism. They can grow both in the presence and absence of oxygen and have a low GC content of 34 to 45%, but have more than 2,500 protected genes. Enterococcus have the potential to survive a different environmental condition of harsh pH, high and low pH, temperature, salinity, etc. So how we can identify enterococcal strains? So initially this enterococcus was placed in the group D Streptococcus microorganism, but in 1986 this was separated and considered a separated genus of enterococcus, and currently more than 80 species have been identified for the enterococcus genus. This is the right side show a schematic representation of enterococcal species identification. For instance, we will select a pink point colonies, then we perform the gram staining biochemical test, and then perform some specific group D test to distinguish between enterococcus and other streptococcus microorganism. Why we are interested in Enterococci? The genus Enterococcus have vast application across different areas, like they are used as a probiotic potential. They are also used in the fermented food. They have therapeutic potential. They also use some genetic engineering and biotechnological applications, enzyme production. They also have the environmental application, which is considered as a fecal indicator bacteria currently in the environments, and they also have the potential to produce bacteriocin, which are called enterocin, and which make them very important in promising microorganism in different microbial fermentation or microbial biotechnological sectors. Although being opportunistic in nature, opportunistic pathogenic nature, a safety and very scheme has been powered to explore their safe nature. For instance, first we will check the antibiotic resistance followed by the virulence vector. If both of them are absent, so then we will go for the production of toxin like biogenic command production can be evaluated, and then other toxin production can be checked. And if none of them has been present, the selected strain can be followed further for probiotic characterization, and if any of them are present, so they may discard for further characterization process. In this research work, this is a schematic representation. We have selected solution from different environmental niches representing different environments. Then we perform the purify these isolates and then confirm their intervocal origin. Then we perform a series of in vitro tests which explore their GIT survivability, addition potential, antioxidant potential, their antimicrobial value, their cell surface properties, enzymatic activity, and safety assessment on different approaches have been elucidated and explored. While in the second phase, we have selected 6 strains which are selected from this pool of selected isolates, and we perform the genomic analysis, which is called a probiogenomic approach, and we extracted the genomic DNA by using CTAB method. The quality of the DNA was being checked by agar-gel, and the library was prepared, and then the next generation probiotic sequence was being performed by the Next 500 genome sequencer at the ICCBS. We have uploaded and submitted the whole genome sequence of these microorganisms to the NCB, and they are being published. Then we perform a series of in-depth genomic analysis to explore their safety, like the antibiotic resistant genes, the virus vectors, and then the biogenetics element finder. We also explore their primary secondary metabolite production, and then we perform some comparative study with the known pathogenic strain. And in the last step, we perform the application studies of these selected interval strains. We have checked their postbiotic production, their tolerance potential against chemicals. We also perform their and check their activity against different enzymes, and their antioxidant potential, DPPH activity have been already explored. And we also check their food-related and biotechnological application, like the temperature effect, the shelf life potential, the yogurt fermentation, and the effect of digestive enzyme on the survivability of these microorganisms. So how and what approach we have used? So these different areas represent the different places, represent the selection point of microorganism we have selected. So initially we have selected 122 interfocal isolate representing the different environments like food, sewage, milk, soil, agriculture soil, cow, air, etc. And they have been explored and confirmed by both phenotypic and genotypic analysis, like followed the basic microbiological approach, biochemical test, and then followed by PCR analysis. And we conclude that more than 60% of the strains are Enterococcus faecium, followed by Enterococcus faecalis, while very few are them are identified as Enterococcus heri species. So we perform a series of comprehensive screening, like for 122 strains. We selected only 11 strains which are completely free from their virulence profile, like gelatinase, catalase, and DNase, etc. We perform the antibiotic resistant genes, phenotypic, plate-based assays, and perform the agar dilution method. So we found only 7 strains were free from antibiotic resistance, but once we perform the genotypic analysis using this trait-specific primer, so we found that 1 strain, although not express the gene and the phenotypic activity, but can harbor that transferable genes, so we discard this strain for further analysis. So we have selected only 6 strains out of these 122 isolates, and consider this as a lead probiotics Enterococcus faecium based candidates, and we further explore for different characterization potential and characterization studies. Initially, we have checked their safety and the form of virulence vectors by both phenotypic plate-based assays and genotypic analysis by PCR have been performed, and we select only those strains which are free from virulence vector. The antibiotic resistant profiling by agar dilution method, diffusion method, and PCR analysis have been performed, and we have selected only those strains which were free from antibiotic resistance. Then there is according to the safety proposed criteria for interfocal probiotic strains. The biogenic amines and biofilm production have been checked by both phenotypic and genotypic analysis by targeting their prime genes. We select and found that only those strains which are free from biofilm formation, hemolytic activity, and biogenic amine production. So then we perform a series of comprehensive intolerance potential against saliva, lysozyme, and pepsin, representing the simulatory gastrointestinal condition, and we found the effectiveness of our selected strain in these tolerance potential. Then we performed we have developed and simulated intestinal and gastrointestinal model, and we have tested our selected strain and found the survivability of these strain in the presence of this stressor condition for 4 hours, and the selected strain show good potential in the presence of these gastrointestinal condition, representing that these strain can be used for application related to the consumption of food or to be added in the food matrix, etc. Once we confirm the genotypic potential or the probiotic characterization of these microorganisms, we perform the whole genome sequencing. This is the very basic workflow which representing the genetic profiling and the probogenomic analysis of these strains. Like we have performed the DNA was extracted, whole genome sequence was performed, and general feature was identified, and then they can compare with non-pathogenic microorganisms. The virulence inspector using the rapid annotation using subsystem technology, RAST analysis have been performed to explore their virulence nature, and we found that only two genes, ACM and EFA, AFM, which are identified in this genome, but we are pretty known that these two genes are involved in the adherence potential of the selected strain rather than the kind of virulence spectra. So again, enhancing their probiotic potential rather than their virulence assessment, virulence profiling. The antibiotic resistance was performed by the CARD tool, like the comprehensive antibiotic resistant genes, and the rapid annotation using subsystem technology tool, and we identified that although the selected strain can harbor only two kind of only two genes responsible for two different antibiotics like gentamicin and tetracycline, but according to literature, these genes are also been identified in many probiotic strains which are even commercially available. So reflecting that these gene make not arise any safety concern in the selected strain. We also check their mobile genetic elements, and we identified in comparison to other selected strains, our strains have less mobile genetic element representing their genetic potential and genetic stability. The metabolite production is considered important properties for probiotics, and we have selected these probiotics using the different online tools and softwares, like the primary metabolites biosynthetic pathways was investigated using the gut smash tool, and we identified only 4 regions are responsible for this primary metabolite production. Then the secondary metabolite production was explored through the anti-smash tool, and we identified 4 regions responsible for these secondary metabolite production. Particularly, the most important is the T3PKS region, which is type 3 polyketal synthase system, which have a very important role in the food system and food science. Then we perform some application-based assessment like antioxidant potential against DPPH, dihydroxyl ion scavenging potential, leferic reducing antioxidant potential, and the ABTS scavenging potential. We identified that the selected strain showed a promising result in the presence of these stressor condition and these antioxidant potential, again highlighting an important their role as anti-inflammatory agents. They perform some functional assays, enzyme production, and then the characterization have been performed, like SLAM production, biofilm formation with Congo red, and exopolysaccharide production, rhodamine degradation, raffinose tolerance, and hydrolysis, and beta glucopyranidase production have been explored by plate-based assay, and our selected strain were confirmed. They have the postbiotic production, and there is absence of any virulence or negative traits in these strains. Additionally, we perform more analysis like lipolysis, dysbiosis assay, twitching motility assay, assays, anti-salmonella activity, stress production, and bioscaling activity have been already explored, and we identified that these strains show good and promising potential, which make them an important biotechnological agent. So, in summary, we say that enterococcus probiotics selected strain have enterococcus probiotic potential because they have survivability in the gastrointestinal tract, they have tolerance to chemicals, they have good aggregation and antioxidant potential, they have good enzymatic profiling, and the selected strain has already been explored for their probogenomic analysis. The selected strain was also tested for shelf life for 3 months, and we identified there was no very less reduction occur after 3 months of refrigerated shelf life storage, and then antipathogenic activity and production profiling of different primary, secondary, and nitrogen production of different metabolites can enhance the replication across food and other biotechnological applications. So the importance and practical implementation and the socioeconomic importance of this research lies that uh, uh, they can help in the development of safe probiotic formulation, strain level characterization to formulate safe, well-defined and topical probiotics for human and animal or other usage. The functional food and nutraceutical industry, as we developed a robust survivability, enzymatic activity, and metabolite production suitable for incorporation into fermented food, dairy products, and dietary supplements. Likewise, this research also paved a way to the clinical and therapeutic application of these selected strains to be performed in the near future. And then there's the industrial and biotechnological application. For instance, they can produce different kind of enzyme and metabolites which highlight and make these microorganisms important agents in different microbial biotechnological applications. While the socioeconomic impact lies because these are a portable healthcare solution, for instance, if you're talking about antimicrobial resistance, so this is currently a very emerging issue across the globe, and the treatment with probiotics or the prevention with probiotics is considered a promising strategy for antimicrobial resistance. So this study can be also explored and can be also targeted for the antimicrobial resistance, and because they are simple and they are a portable solution, even you can perform in the low and middle income societies. They can also lower the reduction in antibiotic usage because they can perform well, and these are natural to the human and other animals, so they can reduce the burden of antibiotic resistance. They can also enhance the local biotechnological industry by when once we investigated their postbiotic protection enzymatic profiling, so they can enhance the local biotechnological sector by development or commercialization of different products. Food security and livestock productivity can be also enhanced by using this research work to integrate with different industry, and we can help in the commercialization of probiotic-based products. In the public health impact, for example, they can improve the gut health and strategies and reduce the healthcare burden and enhance the community well-being. So these can be also used for this application, for this application also. In future, we will perform the in vivo validation study. Although this study connected and integrate the phenotypic assessment, genotypic analysis via PCR analysis, and the probiogenomic analysis performed by whole genome sequencing. But still, we have not performed their in vivo validation study. So in future, we will perform the in vivo study to confirm the survivability and health benefits in animal model and human trials. We can extend this approach to more identification of more probiotic strains from different groups like lactobacillus strains or from other therapeutic strains or some fungal strain, etc. We can also expand the industrial potential exploration. Although the selected strain have potential to produce different kind of important industrially important enzyme, but at industrial level they can be tailored for in future studies. And the socio-economic impact in terms of this to stimulate industrialists and support agriculture and innovative public health using this probiotic approach. So, and finally, this research is financially supported by the seed fund from the ICCBS, University of Karachi, and the Higher Education Commission of Pakistan. To my supervisor, Professor Dr. Saeed Abeli, I would like to say thanks for providing this opportunity and this funding for smooth conduction of this research work. So what is the impact of this research in that form of publication? So we have made several publications and published in the all Q1 journal, international high tier journal, and the highest and the lowest was published in the scientific report very recently. We also publish many review articles which is published in different international impact journals. We also publish several book chapters and different international publishers and highlighting the importance and the probiotic potential of enterococci and other probiotic aspects. Likewise, this work has already been presented in different national and international conferences and been published. Different parts have been published in different international conference proceedings. Is being invited by different different lectures, and as a plenary lecturer, as a distinguished speaker, I delivered different conferences presentation and presented this part of this research work in different sectors and different conferences. So with this, I would like to say thank you from my side. If you have any questions, so you can contact with me through my email. And the Eid, I would like to say thank you for the Global Sensor Award for being again for nominating me for this important award. Thank you.